Hello everyone, welcome back to my YouTube channel. Today we will be exploring the cell-mediated cytolysis and specific topic, that is the perforin and granzyme pathway. So, granzyme and perforin-mediated cytolysis, it is a crucial mechanism employed by the cytotoxic T lymphocyte and another cell, that is the natural killer cells. They perform to eliminate the target cells, okay, particularly those infected with the viruses or transferred into the cancerous cells. So many CTLs initiate killing of their targets via the delivery of pro-apoptotic molecules. Not 100%, but many CTLs. So pro-apoptotic molecules, that is the one type of substances or protein that promote or induce the process of apoptosis, which is also called the programmed cell death. Initially for maintaining normal cellular functions, development and the elimination of damaged or unwanted cells. So always pro-apoptotic molecules are packed within the granules. So there are two types of granules mainly found, that is the 65 kilodalton monomer of pore-forming proteins that called the perforin, and another that is the granzymes of fragmentation, okay, that is a several serine protease called the granzymes. Some CTLPS, okay, cytotoxic T lymphocyte precursors, that lack of cytoplasmic granules and perforin. However, once they activated, they begin to form cytoplasmic granules that include perforin monomers and granzyme molecules. So in picture you can see this after conjugate formation, CTL granules contain granzyme and perforin. So that is bound to be site of interaction, okay, here these granules, okay, that interaction killer and target cell via the activity of microtubules organized center, which I have discussed in my previous video, how CTL kills the target cell. Please watch it. So now these the microtubules which provide to this granules, okay, and the vesicles. This vesicle fuse with the plasma membrane, outer membrane, and release perforin monomer, okay, and granzyme protease into the space at the junction between the two cells, that is the synapses. Cell mediated pore formation in target cell membrane. Once the granules release their contents at the synapses site, okay, this is the CTL cell, cytotoxic lymphocyte, and target cell. This is the membrane of CTL, and this is target cell membrane. Okay, granules, they fuse with the plasma membrane and exocytosis through the release of perforin monomers and granzyme. So in this model, okay, arise in intracellular calcium triggered by the CTL target cell interaction. The first step, induce exocytosis in which the granule fuse with the CTL cell membrane, okay, calcium dependent, that is the, and release monomer. Second stage, that is fuse with the plasma membrane, and the release of third, it is the release of perforin monomer undergo calcium induced conformation. Okay, change they allow them to insert into the target cell membrane. Here it is. In the presence of calcium, the fourth step, okay, the presence of calcium, the monomers polarize within the membrane and forming cylindrical pores. That is a side by cylindrical pores, okay, that is entire completed pore and polymerized perforin. It is also one of the picture here for the formation of pore into target cell membrane. Okay. So it is the allopathic monomer, okay, allopathic chain inserted into the target cell membrane. Now the granzyme B. Although granzyme B is once thought to entry into the target cell by the surface perforin pore, and it's now thought to enter into via the endocytic process. Okay, so this is the perforin pore, and this granzyme inserted into the endocytic process to the target cell. So the many target cell have molecule known as the mannose 6-phosphate. Here it is a mannose 6-phosphate receptor in the surface. It also binds to the granzyme B. Here is granzyme B, which binds to the this one and internalize to the and appear inside the vesicle. This is a vesicle. Now the perforin internalize at the same time. Okay, and the forms a pore that release granzyme B from the vesicle into the cytoplasm of the target cell. This is a form of the vesicle, and they both are the into the vesicle granzyme B, which is the form into the vesicles. Okay, so this is also one of the picture here. That is the natural killer cells. They also kill the target cell by the granzyme and perforin pathways. Okay, degranulation, lipid shield. It is the cell membrane, presynaptic membrane, synaptic left, postsynaptic membrane. Here it is. Perforin pore formation. Here is the perforin that form the pore. Granzyme B entry and cell death. Okay, so this is the pathway of perforin granzyme. Granzyme B perforin, once they enter into the target cell, they start the signaling and create the apoptosis of the cells. Okay, so perforin, the entry of the granzyme B, exit vesicle and perforin dependent process. BID that activate the BID, and BID that also activate the mitochondria, and mitochondria that release the cytochrome C, which binds to caspase 9 and APA-F1, and this inactivate the pro-caspase 3 to caspase 3, and this caspase 3 is initiate the apoptosis. Okay, apoptosis substrate and active apoptotic effector and apoptosis of the cells. And also granzyme B directly that they activate the caspase 3. Once the granzyme B inserted into the target cell, that initiate the cascade of reaction that result in the fragmentation of the target cell DNA into oligomer of 200 base pair. This type of DNA fragmentation typically apoptosis, and which within the 5 minutes of CTL contact with the target cell begin to exhibit the DNA fragmentation. Okay, so this apoptosis process that does not require mRNA protein synthesis in either the CTLs or the target cell. Now the one of the more mechanism, that is the how do CTL protect themselves from their own perforin granzyme activity. So CTL possessing the one of the most important protein, that is the serpins. The serpin that is the protecting CTL from their own pro-apoptotic function or pro-apoptotic molecules. And one study showed that the CTL don't survive in mice deficiently for one of the most common serpins expressed by the CD8T cells, or which is called the SPI. Okay, some are the MCQ based questions here. Okay, this is all about today's video. Solve these questions and answer this question into the comment box. Thank you for watching.